recruitment for the Strong Heart Study, 4 states in the Western US, including Arizona, Oklahoma, and both North and South Dakota. And we can see on the right that each participant that we have information on participants from multiple study visits. So the first study visit on the right was actually in the late 1980s, from 1989 to 1991. We can see that there are multiple visits over time. What this means is that we have multiple time points for each participant where we have information. Also, importantly, we can see that there's this ongoing surveillance of morbidity and mortality information. We can leverage this to look at how different metal exposures are related to cardiovascular disease. And just to put this in context, these are real people, and I just want to highlight all the great participatory science and community that's involved with the Strong Heart Study. So this is a picture of community members and researchers from the Strong Heart Study that worked all through COVID. You can see everyone is wearing masks. And it's really been an opportunity to be a part of, and it represents truly amazing cooperation between community members and scientists that I've been lucky to be part of. I also had the great opportunity to visit South Dakota only twice, but I was able to learn a lot more about what the community members really value and how our work fits into all these different priorities. So it's been a really rewarding experience to be part of the Strong Heart Study. I also want to get everyone up to speed on what the Strong Heart Study is. So this is some basic descriptive information about the cohort. I want to highlight a few things from this slide. Firstly, the age of participants on the original baseline recruitment was about 55 years old. So this is generally an older population, hence why we see a lot of cardiovascular disease, clinical cardiovascular disease outcomes. I also want to highlight that there's a high prevalence of diabetes as well in this population. So we can see that the prevalence from this first study period for one of my analyses was about 42% of people had diabetes. So again, this is a population with cardiovascular disease and diabetes that there needs to be a lot more research and attention in these communities to solving these problems. I also mentioned that the Strong Heart Study has a lot of information. So there's a lot of data collection concerning metals that have taken place over many decades, actually, at this point in the Strong Heart Study. I'm going to walk you through how we can actually measure metals at different time points to connect with these heart disease outcomes. So in a few slides, I'm going to emphasize how we have uranium measured in urine at the 4th and partially the 5th visit, how we're going to connect these to subclinical measures of cardiovascular disease. I'm also going to highlight that we have lead collected at multiple different time points that we can connect to clinical cardiovascular outcomes and also understand how lead changes over time to connect to improvements in cardiovascular. I also want to finally mention how we also have DNA methylation data in blood. We can use this and epigenetic clock to generate epigenetic biomarkers of lead exposure and ultimately connect these to health outcomes. We have a lot of different time points and a lot of different information on metals that we're going to leverage here and connect to different cardiovascular endpoints. So with that, I want to get into the first part of my dissertation concerning how do we assess levels of metals both in the water that we drink and in our bodies. And this part has 2 objectives accordingly. So the first objective is, can we use publicly available data to get an understanding of what Strong Heart Study participants drink in terms of uranium present? So again, how can we use this nationwide uranium data to make really good estimates of what the concentrations of uranium are for these participants? And secondly, now that we have a measure of uranium in water, we also care about what uranium is in someone's body. So how does the relationship look between uranium measured in water and uranium measured in someone's body? Ultimately, can we develop a model to then predict what uranium is in urine based on what we have in water if we do not have uranium estimated in urine? So that's where we're headed. But before we get into the specifics, I want to give a few slide overview of what uranium is and why we should care about it. So many of us, we've all probably heard of uranium. It's actually naturally occurring in many rocks and soils throughout the United States, especially the Western United States. Uranium is radioactive, but actually we're more concerned with the chemical toxicity of uranium rather than the radioactive toxicity of uranium. So for this work, we're really focused on the chemical toxicity in relation to heart disease. And uranium, it's related to a variety of adverse health effects. I'd like to emphasize that there has been limited research performed on how uranium relates to heart disease, which is another reason why we need to study it. And also for the chemical toxicity, we're primarily focused on how someone ingests uranium through their drinking water and also diet, but mainly through drinking water. This is the main exposure route that we're interested in. And because of this, I want to highlight how uranium is very widespread in water throughout the Western United States. In fact, it's detectable in over 50% of public drinking water systems. And here we have a map of the Western US. And this shows us at the county level what the concentrations of uranium are in water, with darker shades of color indicating higher concentrations. And I mentioned that the Strong Heart study recruits from 4 states. These 4 states are highlighted in these blue boxes. And the takeaway here is that we see that uranium is a concern in public drinking water systems. So we really need to be paying attention. Further, we'd be remiss to mention not to mention the legacy of anthropogenic mining, milling, and other forms of uranium processing that has taken place both directly on or in areas proximal to American Indian communities. So this is a map of uranium activity locations in the Western US. We can see that uranium from mining is a real problem in these communities. And essentially what we're trying to do for the first objective was, can we estimate and assign water uranium from public drinking water to participants? The answer is yes, we can do that. And this is how. So thanks to Annie's group and the Water Epi Lab, they've worked to refine these estimates that use publicly available data from the United States Environmental Protection Agency. And this results in very fine estimates at the zip code tabulation area level, which is a very fine spatial unit. And what we can do with this is say, we can get an understanding of what the uranium is in water for a strong heart study. So below is a map of South Dakota, again emphasizing the varied, but in some cases, high burden of uranium in the water for these participants. And I want to highlight the flow chart of how we actually linked uranium concentrations from publicly available data to participants. So we were able to get these estimates from data, and the Strong Heart study was able to know where each participant was. So that's not information that I'm allowed to know because it's private, PHI, but they were able to make a linkage for us and actually generate estimates for each participant. There's some nuance in these estimates, but for one of my analyses, we found that there was a mean of about 4.5 micrograms per liter of uranium in drinking water. So again, demonstrating that this is a concern. The second objective, as I mentioned, was how do we go from water to urine in terms of uranium concentrations? And I just want to highlight that this is really important if we're trying to determine relationships with health effects and safe drinking water standards. So ultimately, we need to know what concentrations are in water, how they relate to concentrations we measure in the body. That's how we use urine as a measure of internal dose. And ultimately, what does this mean for health? So we need to know both of these time points, both of these instances rather, both in water and urine, to then relate this to health outcomes. And as I mentioned, if we could develop a prediction model to go from water to urine, we could essentially estimate urinary levels where we don't have this data available. This is a really valuable resource. We're trying to get an understanding of someone's exposure on a population level. So with that, I'm going to want to again highlight that we're going to use uranium in urine to go from water to urine concentrations. And in our prediction models, we went over this a few times, but we were trying to understand what types of information could be useful for us to predict a urine mixture. And what we sort of arrived on is we need to think about prior knowledge of what contaminants, both in water and urine, that are relevant, including contaminants that may coexist with uranium. This can give us information about what do we expect a high or low concentration in someone's body based on what we see in the environment. And again, figure out different sources of uranium. So ultimately, what we found and the important variables for our prediction models are highlighted. We ultimately found that concentrations of other metals present both in the water and in a person's body are really important at getting at this relation between water. This includes obviously uranium in water, but also things like arsenic and selenium, both in water and in the air. Also want to highlight that we considered other sociodemographic variables and clinical variables, and we found that study center may be approximating some changes across center of recruitment, as well as urinary creatinine, were really important for our predictions. These variables made us have better predictions of urinary. And to give a quick highlight, we also wanted to see how well are predicted values of urinary uranium compared to actual measured real values of uranium. This is what these two plots show. So on the left, we have a scatter plot of the predicted values versus the real values on the y and x axis accordingly. And the closer to this line, closer to the one-to-one relationship, that we would expect, or we would be seeing a better prediction model. On the right, we have two curves, same idea. One's the predictions, one's the actual response values. The predictions are in blue, the response values are in green. And the takeaway from this curve is that if these two curves were exactly the same, that means our predictions would be perfect compared to the actual values. We see that they're not perfect, but they're still pretty good. Even though the height of the peaks is not the same, and maybe at the tails of the distribution, there's not as much overlap as in the center, we do actually make pretty good predictions of urinary uranium values. And the takeaway here, again, we're trying to understand, for uranium specifically, what it looks like in the water and what it looks like in the body. And it's really cool, and I want to highlight that we're doing this with very fine resolution data, so at the Zicta level. And this is really novel, and this is really important for more accurate exposure. Not to say we can't further refine these prediction models. We most certainly will over time. But regardless, this has really big implications for exposure assessment, environmental monitoring, as I mentioned before, connecting these levels to cardiovascular health. Okay, so that was one part about uranium in the environment and in our bodies. I then want to go through how we can connect these measurements to health measures. So in the Strong Heart study, we have a lot of different information about cardiovascular disease. And firstly, I'm going to talk about how uranium relates to subclinical measures of disease. So if we look on the bottom here, this is a progression of a disease. We have an exposure on the left. And after we have this exposure, if we have disease, we see subclinical disease accompanied by pathological changes. And importantly, this is usually before the onset of symptoms and the time of diagnosis. And also, this is before you get clinical disease. So it's really, really important to understand and intervene on subclinical disease, which reflects early stage cardiovascular disease in this case, compared to clinical disease, where it may be too late to intervene. So accordingly, if we're interested in subclinical disease, we were able to leverage these echocardiographic measures, which give us really good information to understand subclinical disease. And ultimately, our objective for this part was to understand how uranium is related to these echocardiographic measures. And what I mean by this is, how does uranium relate to measures of cardiac geometry, functioning of the left ventricle, and the presence or absence of left ventricular hypertrophy? So let's just take a second to actually explain what each of those outcomes are. So in the Strong Heart study, they have these echocardiograms, which is essentially using ultrasound to take pictures of the heart. There are different types of outcomes that we're investigating as well. All the way on the left, we see that cardiac geometry is one type. This includes things like diameter and thickness. This is a physical measure of your heart. From these measures, we can get an idea of what someone's mass is of the left ventricle, which is the most important, very important chamber of your heart. And from this mass, we can also get an idea if someone has left ventricular hypertrophy, which is characterized by an enlargement of the left ventricle and a thickening of the walls, which generally puts more strain on your heart. There are also functioning outcomes that we considered, which considered measures of blood flow, things like the amount of blood pumped in a given cardiac cycle. But we did not have enough time. It's too much already, so there's not enough time to talk about these functioning outcomes. So primarily, we're just going to be interested in the geometry and hypertrophy outcomes. So again, I want to highlight that we have uranium measured at a very specific time point, and also these echocardiograms measured at two different. So importantly, at visit 4, we had information on urinary uranium from participants and also information from these echocardiograms. We also have echocardiograms taken at visit 5 in the future, which is a really powerful time point if we're trying to connect exposure that happened in the past and what you see on an echocardiogram today. Very high level, this is how we set up some of our models. So the main exposure was uranium measured in urine. We used linear mixed effects models. And in this example, our outcome was left ventricular hypertrophy. We had a fairly large sample size, about 1,300 people. And I want to highlight that we both adjusted for sociodemographic and clinical characteristics to get a more true understanding of the relationship between uranium and these cardiovascular measures. Also want to highlight that these are subclinical outcomes. So we'd expect to see these hopefully, well, not hopefully, but we would not expect to see clinical outcomes in a younger population. But for this study, the sample size, the mean age was about 31 years old. So this is a younger American Indian adult population where we may expect to see changes with uranium and subclinical disease. And so with that, I want to highlight the main results from this analysis. So in terms of words at the top, we saw that uranium was related to increases in left ventricular mass. We also saw that uranium was related to increases in left atrium diameter. And these measures were really important, mass especially, to get an understanding that uranium was related to an increased odds of the presence of left ventricular at this follow-up time. That's a very, very important finding. The graphs below show how uranium relates to both these cardiac geometry outcomes. Uranium is on the x-axis and increases from left to right, and the left y-axis shows the increase in each cardiac geometry. So we're looking at left ventricular mass index on the left atrium diameter. And essentially what we see is that these lines are increasing there, going from the bottom left to the top right. That means essentially that as uranium increases, we also see increases in the left mass index and especially the left atrium diameter. There's two colors on these graphs. This corresponds to the 2 time points that we have these upward. And if you look especially with left atrium diameter, there's a very consistent increase as we go from higher uranium to an increase in left atrium diameter. And all this to say is it suggests there may be a link between uranium and early stage cardiovascular disease based on what we found. And this is really important work to be doing in young American Indian adults. So it's really important to understand what the impact of uranium is. And potentially how uranium may be related to other subclinical outcomes or even clinical outcomes, especially over long time periods of exposure. That being said, there is a great need to do replication for this analysis in both American Indian and additional populations, and to ultimately have efforts to reduce uranium exposure. Okay, now I'm going to switch gears into talking about lead, and also switch gears into talking about clinical disease. So we just went through how uranium is related to subclinical disease. Now I'm going to talk about how we can use different measures of lead to relate to clinical cardiovascular disease. This is really important because we can leverage the full study timeline of the Strong Heart study, have a lot of power and have a lot of information to look at relationships with disease. In this first part with lead, we're going to talk about how simple blood lead measures relate to these clinical incident and mortality outcomes. And secondly, I'm going to take you through how we can use epigenetic biomarkers to also look at these clinical. And I'll say it right now before we get into it. We found great consistency when we looked at lead measured in both of these ways with our outcomes. In other words, we found that lead was associated with increased risk of cardiovascular disease. But in terms of the background, I think we're all familiar with lead a little bit. Hopefully we're aware of the great public health victories that have included bans on lead in paint and gasoline and other things like that have resulted in declines in blood lead levels over time. But that being said, environmental lead exposure globally is still a huge concern. It was estimated that this exposure is linked to about 1.5 million cardiovascular disease deaths in 2020. This is a huge global concern, and it's actually still a concern in the United States communities as well. Most importantly, we really do more work about understanding lead and what levels relate to disease, especially in American Indians that have high levels of metals exposures. So for the first part of the lead work, I want to highlight that we're going to use blood lead data collected from the third Strong Heart study visit, which took place from 1998 and 1999. And again, leverage the entire timeline of the Strong Heart study, which continues to the present day, to look at relationship with clinical, incident, and mortality outcomes. So this is how we set up our models. We looked at blood lead as our main exposure. We used survival analysis to look at both incident cardiovascular events and cardiovascular disease mortality. And I want to highlight that we, again, adjusted for relevant sociodemographic and clinical characteristics. And just to put these numbers in context, for this analysis, we saw that about 1/3 of participants suffered a cardiovascular event, things like stroke, for example, and about 23% of participants suffered a cardiovascular disease mortality. This is a very high burden of disease in these communities, and it's really important to look at levels that relate to disease. So that's what we did. These plots are set up in the same way as the rating plots. Each square here is a different type of outcome, and I'm going to mainly walk you through one of these to make it simple. We're going to look at overall cardiovascular disease mortality. The takeaway from this plot and this analysis that we did is we found that increasing levels of lead were associated with an increased risk of overall cardiovascular disease mortality. And this plot below shows that we have lead levels on the x-axis increasing as we go from left to right, and the hazard ratio on the left y-axis showing that as lead levels increase, we do see an increased cardiovascular disease. I want to highlight that there's an important inflection point on this graph where the line really shoots up, showing that if you have above this level, your risk rapidly increases. And that level is about 3.5 micrograms per deciliter. I don't think it can show up here, but about 3.5 micrograms per deciliter, or 35 micrograms per liter, we see that there's this risk of CVD mortality that shoots up. So that's an important level to pay attention to. Yeah, some of the takeaways from this analysis, I mentioned that this was important to do in American communities, and we did emphasize that lead is a cardiovascular disease risk factor. But I also want to highlight that the new CDC blood lead reference level, it was 5 micrograms per deciliter, and they recently lowered it to 3.5 micrograms per deciliter. The idea was that they were trying to capture lower levels to get more children into care. But this level is really important, set by the CDC, to pay attention to, including how does levels above or below above this mark relate to cardiovascular disease. So we found that there was some consistency around this measure and eclipsing this measure with increased cardiovascular disease. Ultimately, however, this really speaks to how we need to reduce lead exposure across all communities in the United States, including and not limited to American Indians. The second part of the lead work, we're trying to estimate, again, the relationships with clinical cardiovascular disease outcomes. And I mentioned how we had blood lead before. But if some of you have learned about lead, you may know that bone lead is actually the best marker of chronic exposure, chronic and cumulative lead exposure. And this information, we do not have it in the strong state. So I'm going to walk you through how we use these DNA methylation-based biomarkers to generate estimates of lead, both in blood, but also in bone in the strong state. And ultimately, how we're going to use these estimates of lead to relate these to clinical outcomes. So I mentioned blood lead before. This is a great biomarker for recent lead exposure. It has a much shorter half-life than bone lead, but a half-life about 1 to 2 days. Bone lead, however, can capture this chronic and cumulative lead exposure, has a much longer half-life, about years. The problem on a population level is that it's very expensive and invasive to measure bone lead for people. You have to essentially at 2 sites in the patella and tibia are common sites. You have to essentially sit still and take x-rays of a person for about 4 minutes. This is not really feasible to do on a population level. And in the Strong Heart study, we do not have bone lead available. But luckily, we do have data on epigenetics available. I'm going to walk you through how we can use epigenetics to help estimate these levels. So as a primer, DNA methylation is one type of epigenetic marker. And epigenetic markers, including DNA methylation, are sensitive to environmental exposures. DNA methylation specifically is the attachment of a methyl group to certain points along the DNA sequence that does not change the sequence of DNA itself. And these are really important for growth and development, but we've also seen that metals are associated with DNA methylation. This can have profound impacts on you. So essentially, a group at Mount Sinai and Columbia leveraged that metals like lead make these DNA methylation changes in people. And we're able to construct biomarkers of both blood and bone lead based on these changes that lead makes. So we wanted to ultimately apply these methylation-based biomarkers in the strong heart study. But firstly, let me show you how they actually made these. So again, yeah, in the strong heart study, we do not have these measurements available, but we're ultimately trying to. So from the previous study, I want to highlight that they had DNA methylation data available in blood. They had this bone lead actually taken from participants in both the tibia and patella. They were able to use elastic net to construct biomarkers based on the methylation changes associated. What they found, and this is the picture on the right, is they found that there was good correlation, relatively good correlation between both the measured and estimated concentrations of lead in bone and what they estimated this to be. And this was really encouraging for us. So we saw that the guesses of what lead was corresponded pretty well to what the actual measures of lead was. So in the strong heart study, we then wanted to leverage the fact that we have DNA methylation data available to estimate lead levels both in blood and in bone for this population. So this next portion is going to be highlighting how we had DNA methylation data measured the first visit, which is really important because if we can estimate lead this first visit, we have even more of the timeline of the strong heart study connect lead exposures to these clinical events. So with that, we were able to successfully estimate these lead exposures, and the concentrations in both tibia, patella, and in blood are at the bottom right. But we also wanted to look at how these related to clinical events. So we set up our models in a very similar way, where our main exposure was individually, each type of these epigenetic biomarkers of lead. We used survival analysis with the large sample size to again look at clinical cardiovascular events. Again, we adjusted for relevant sociodemographic and clinical characteristics. And the main result from this analysis is this slide. So the takeaway is we found an increased risk when we looked at lead measured in both tibia and blood epigenetic biomarkers with cardiovascular disease mortality. And the hazard ratios are highlighted in these pink boxes. But essentially, we found that these estimates of lead, both in bone and blood, can potentially capture the cardiovascular impact of both cumulative and recent lead exposure. So this was a really profound finding for us because we found that even though these biomarkers were created in a different population, we're able to apply them to the strong heart study and still see strong relationships with cardiovascular disease mortality. And this is really useful. You know, this is a non-invasive way to get an understanding of what lead exposure is for a population and get at this measure of bone lead, which is the best measure of chronic and cumulative exposure. Importantly, because we did not have bone lead available in the strong heart study, we were not able to validate these measures. So future work using these epigenetic biomarkers really needs to validate these measures and explore them more given their great potential. Okay, so we just went over two big parts of the dissertation. The first part was trying to understand how can we know more using publicly available data about what uranium is both in water and urine. The second part was relating these levels of uranium to subclinical disease and led to these clinical outcomes. But now I want to show you what happens if we reduce exposure. So if lead levels, for example, decline over time, what does this mean for cardiovascular? I'm going to start with showing you relationships with subclinical cardiovascular disease outcomes, with the clinical effects maybe being sometime in the future. So the main objective of this third part was to understand, do declines in blood lead concentrations over time result in lower blood pressure levels? Or said another way, do declines in blood lead result in improvements in blood pressure? And so this was recently published. We found that, yes, these declines do improve blood pressure. So this was recently published and very well received, actually. Mostly Annie, but I was there too, did an interview from the NIH, and they really responded well to the fact that when you reduce levels, you improve health. This was a nice change from the usual saying, if you have a high level of a metal, you're going to have a bad outcome. This perspective and this really shift to like an improvement on an intervention was really useful, I found. And just again, so the timeline of specimen collection, we had lead measured at 2 different time points. And that's how, for each participant, we knew if there was a decline in lead over time. We had two measures for each participant on both lead. We had two measures for each participant on blood pressure. So based on that, we could calculate both a change in lead over time and a change in blood pressure. And we set up our models as such, where we looked at this decline in blood lead, how does lead change over time with changes in blood pressure. We used generalized estimated equations and a sample size of about 300. Again, adjusting for relevant personal and clinical characteristics. From here, I want to highlight that at baseline, the baseline lead levels was about 2 micrograms per deciliter or 20 micrograms per liter. And over about a decade, so in between visits, each participant's lead level, or on average in the population, lead levels declined by about 33%, to about 0.67 micrograms per deciliter or 6.7 micrograms per liter. So this is definitely a good thing. We've seen that lead levels go down over time. But how do we relate that to health outcomes? So this is one example of blood pressure, and this is the main finding of our research. We looked at how declines in blood lead, which is now on the x-axis, relate to changes in systolic blood pressure. And what we see is that as blood lead levels decline, so as this number goes from left to right, we see that there's a really sharp drop in systolic blood pressure. But what this means, to say it another way, is that at about 0.1 micrograms per deciliter, or 1 microgram per liter, we see that all of a sudden there's this really steep drop off in blood pressure. And the interpretation here is that whenever we see that a participant had a decline in lead over time, we see that the blood pressure did decrease, with larger declines in lead over time resulting in larger changes and reductions in systolic blood pressure. So this is a really important finding because it shows us that even when declines are low and when lead levels are fairly small to begin with, we see these improvements in blood pressure, systolic blood pressure. And this sort of begs the idea of what happens if we do this work in other populations that have slightly higher blood lead levels to start? Do we see even further reductions in systolic blood pressure, even further improvements in cardiovascular health? With that being said, we did perform this work in a relatively small sample size, about 300 people. So it is important to do more work on this topic. But despite that, this takeaway here is that this is really great evidence of further reducing blood lead levels. Again, even when these levels are low, we saw that if we reduce these even further, we see improvements in systolic blood pressure. Great. So to talk about conclusions, I want to remind us where we began the talk. So this work really entered in the gap where there's this under-recognized relationship between how metals influence cardiovascular disease and cardiovascular disease risk. And really, there's a need to recognize how metals are cardiovascular disease risk. So hopefully I convinced you that we need to care about metals and cardiovascular health a little more. Again, by looking at how are levels of metals in the water relating to levels of metals in our bodies, and what does this ultimately mean for health? We've also seen that when we reduce these metals, we've seen improvements in health. So hopefully you're convinced that there needs to be more research in this area. But just very big picture, high level from the first part, I want to remind us that these publicly available Dicta level estimates exist for many contaminants, including uranium, in public drinking water systems. And we were able to leverage these to actually predict what a urinary measure of uranium is for us. And as I mentioned before, this has really wide applicability to public health intervention and health systems. It's really important to go from water, urine to health effects, and really get an understanding at what levels in water are safe for people to be drinking. Ultimately, if we can accurately predict a metal in a population from using water data, this has applicability to other contaminants. So hopefully this work paves the way for future relationships between water and internal dose measurements for other metals. I'd also like to highlight from the second part the important need to pay attention and ultimately intervene on subclinical disease. So I showed you that uranium may have these relationships with subclinical disease. Really important to intervene before someone develops clinical disease that presents with an outcome. Furthermore, we've seen how important it is to look at lead at low levels that relate to disease, and ultimately reduce lead exposure even at these low levels. And I'm highlighting again how hopefully my work and other work can integrate both more traditional biomarkers of metals exposures and epigenetic biomarkers into environmental health questions. Because hopefully I've showed you that epigenetic biomarkers can be very, very useful to allow us to assess exposures that may not be possible to capture in traditional biomarkers. For example, we did not have bone lead measured at the first visit of the Strong Heart study, but by using epigenetic biomarkers, we're able to estimate bone lead and complement how we can assess lead in 3 different types of tissue and relate these to clinical disease. Lastly, we talked about how declines, even when lead is very low, can provide relatively short-term benefits for cardiovascular. I mentioned the CDC reference level. We've seen that even at low levels, we do see improvements in blood pressure, for example, when we reduce lead even more. So it's really important to still pay attention to lead, even though we've all heard about it for a very long time, even at low levels. And honestly, to do this work even further in other populations that have maybe higher lead levels, do we see even larger improvements in blood pressure? And ultimately, connecting this to the need to have more interventions to reduce lead overall, because we've seen that there are many benefits with lead. And so this is my last slide. In terms of future directions, if we really care about metals and cardiovascular disease, going forward, we really need to acknowledge the role of climate change. So not only will people change across the globe and migrate, but the spatial concentration and distribution of metals will also change. So if we're thinking about how people are exposed, maybe in drinking water, what does this exposure mean for a person and connect this to health? We have to acknowledge how climate change will essentially change how metals, how we are exposed to metals. We can also think of differing exposure through this transition to renewable energy and electric vehicles. We have a lot of different people being exposed to metals in different ways. And if we continue pursuing technology that relies on metals, this is something that must be acknowledged if we care about health outcomes. And then at the bottom here, to sort of anchor where we started, all of my work has been done in the Strong Heart study. And I've showed you that there are both adverse connections with cardiovascular disease and hopefully with cardiovascular disease we reduce metals exposure. But we really need to pay attention to conducting this research with communities and ultimately protecting and advocating for these communities that have high levels of metal exposures. These are the communities that we need to be understanding and asking these questions in to ultimately reduce exposure. So with that, I want to acknowledge my phenomenal mentoring team of Allison, Anna, Annie, Dr. Ben Bostic, Dr. Jason Human, and Jeanette, who have been lovely for my F31 and the prediction models. Also want to acknowledge all these Strong Heart study participants, investigators, researchers, community members, Missouri Breaks, everyone who's been involved with this work, because without these people doing the hard work, I would have nothing to present on, quite honestly. I also want to acknowledge the entire EHS community, all the different lab groups that have listened to me present this work many times, the metal lab that helped analyze the more recent metal estimates, all the doctoral students, both past and present, that have worked on this in these areas before and paved the way for our research, in addition to all the collaborators and colleagues that have been part of the last 4 years for me, and prior to that as well. I acknowledge all my family and friends that have come out, especially my beautiful wife, Stephanie, for all her support. And I want to also acknowledge all my different funding that has helped me over the years. So thank you so much. Oh, yeah, right. Yes, so we looked at those measures of geometry and functioning as well. I think the only interesting finding we saw was changes in interventricular septum thickness. The full list of outcomes is in the paper, but the main finding really was with blood pressure, especially stop. Um, well, they're both toxic and the distribution is, I guess, similar. So uranium does store in the bones as well. It's stored like lead. But primarily we are we're concerned with kidney toxicity for uranium. Um, it is interesting, if this is the direction of your question, if we can use epigenetic biomarkers to estimate levels of uranium in bone. But we relied on the fact that urine is a great measure of internal dose and a good biomarker for uranium exposure. Um, that's an interesting idea. Yeah, it's interesting. I can say that I don't fully know why that is. But my yeah, I'm not sure biologically why we would see it at that specific threshold. Um, but yeah, that's an interesting question. Yeah, I don't know if we looked at specifically levels that corresponded to that change with 3.5. The baseline lead level was about 2 micrograms per day at the start. It did decrease by 33%. But to your other point, as we saw larger declines over time, over about 10-year period, we did see correspondingly lower uh, improvements in blood pressure. So the improvements did increase as we saw larger improvements. Definitely, yeah. The original recruitment was an older population. We have seen these lead levels decline over time. And in the younger population of the family study, that's where we have the baseline level of about 2. Right. Thanks. No questions. Yes. Yeah, so to answer your first question, so there has been recently more cooperation and acknowledgement of lead in these communities and efforts to reduce exposure. But mostly, we think that the same reasons for the lead declines that we have seen in the US population are applicable for the lead declines we've seen in the stronghold area. So phasing out of lead from gasoline, paint, other consumer products, um, and a greater understanding around lead in potential food and things like bring lead into the home. And all those efforts have resulted in these declines. Thank you.